that's going to shed its skin? Except reptiles. I don't think that's chitin. Reptiles have chitin? I'm going to have to look that up. I do not believe that outside. I think it's for them, it's keratin and not chitin. So there's a cicada that is shedding its skin. This is what chitin looks like. The only difference is it's still the same thing. An oxygen, 1, 2, 3, 4, 5 carbons. So we still have all of our functional groups on the outside, but now down here, what we've added is this nitrogenous base, this base that has nitrogen in it. It's a protein is what this is. And by adding this protein to this carbohydrate, now we have this different form. And chitin can come in 2 different forms, I guess is the word I'm going to use. So it can actually be soft and pliable. If you've ever seen it when a bug is getting rid of its skin, spiders do this. Tarantulas get rid of their skin. This is what's going to come off of them, and it can be really soft as these combine with each other. So as each of these polysaccharides and each of these proteins bind with each other, it becomes like stronger, if you will. We actually use chitin in surgery, so it can be very strong. We've already bound them together in this point, and then what's going to happen as it's in your body? It's organic. It's a polysaccharide. It's a bunch of sugars and a protein. What's your body going to do to it? It's going to break it down and just reuse all of these components to make something else in the body. As it does that, that means the stitches now dissolve inside your body. So that gives us strength, and then we don't have to, we can do surgery in layers and have the stitches on the inside doing these different layers of stitching, which actually has increased the efficiency of plastic surgery. Now scars don't have to be as big or as clunky because we can go in and use this, and knowing that we don't have to pull those stitches back out. We don't have to damage the tissue again. All right, next one, is our last one for this, is lipids. So lipids are... these do not form polymers. These guys stay on themselves. They are, if they have no affinity to water, we call them hydrophobic. So this means they do not like water. If they do not like water, generally they are non-polar. If you remember from chapter 2, polarized molecules are ones that have an affinity for other molecules' charges. So water, H2O, wants to grab onto other waters, and then other waters, and those hydrogens and oxygens are having interactions with each other, which is why water, one of the reasons I should say, not all, but which is why water stays in a stream. So water generally stays together because of that polar bonding. Well, water is polar and lipids are not, and therefore the lipids being nonpolar don't attach themselves to water. So all fats and water don't mix. If you're trying to make a salad dressing or you put a little bit of oil with your pasta, they don't mix together because they're not meant to mix. The one's polarized and attaching itself to each other, and the other one doesn't want to attach to everybody else. So fats always have a glycerol. So this is a 3-carbon alcohol. That's the glycerol that's on the top. With the hydroxyl group. So here's our glycerol, and then we're going to have a hydroxyl group where we're going to have these ester linkages, and this linkage right here, you're going to have 3 sets of links. So here is your glycerol that's on the top. If you look in most of your lotions, so most of your lotions, second or third ingredient will be glycerol, if it's a really good lotion. So glycerol adds fats to your skin, which makes it softer. So here's the ester linkages right here, 1, 2, 3, and they're all linked to a fatty acid. The fatty acid is non-polarized, so it has no spot where it has extra energy, where it's positive or negative. It's all balanced, and therefore will not interact with water whatsoever. And this is why fats don't interact with water. So a fat stays on its own. It doesn't want to mix with water. So as the water tries to gather together, the fats get left in these clumps because they're not polarized with the rest of the water. So that's what we're looking at. This is our connection that we're going to end up here, our linkage that we're going to end up with each of these fatty acids. We're going to do this dehydration reaction once, twice, 3 times, three times, so that we end up with this fat molecule that has this large head at the top and then has these long chains that go on the bottom. Count the number of carbons: 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14. So this is 16. I know if you've ever seen the movie Lorenzo's Oil, ALD, aldoleukodystrophy, is caused by having too many C24 fatty acids, and that is a chain just like this with the glycerol at the head, but the chain is 24 carbons long. So the body can't get rid of those, and they had to find a way to make the body get rid of those. So we just talked about that. And in the fats, because you have this glyceride, so we have a glyceride, and we're adding it together with our 3 fatty acids, we call it a tricycle glycerol. So glycerides or glycerols, either one, are because there's 3 fatty acids with 3 glycerols at the head, at the top of it. Okay, so here is, hopefully this has really good connections for you. By the way, as far as glycerols go, there are many more glycerides or fats in female skin than there is in male skin. So women tend to have more fatty acids in their skin, which tends to make your skin a little bit softer. Okay, so now we're going to go into saturated fats and unsaturated fats. This is big for your This is big for your diet. So everybody's heard of saturated fats, how they're bad for you. Unsaturated fats are good for you. I'm going to explain to you why they are good for you and why they are bad for you. Okay? All right, so saturated fats are, this is the bottom line, just breaking it down. Anything where this carbon chain down here, so here's our glycerol at the top. So anything where this carbon chain at the bottom is saturated. Saturated meaning that it has every single carbon down here has hydrogens attached to it, so they have all 4 bonds used, and they're all single bonds. So that is important. Being all single bonds, pardon me, they can lie right next to each other. They literally can get right in there and cozy up, and you end up with layer and layer and layer and layer and layer. And so even at room temperature, saturated fats are solids. There's no room in there for them to become a liquid and to have spaces because they have this structure. So all saturated fats are solids at room temperature. That would be butter, any animal fat. If you've ever made a steak, so if you've ever made a steak and it sat out, and you left it sitting out and it got cold, you'll notice that the fat turns white on the bottom of the plate. So that white part is because it is an animal fat, and all animal fats are saturated fats. So anything that comes from an animal as a fat should be solid at room temperature. If you heat it up, then you're going to break apart each of these rows. And as you break them apart, it becomes more liquid. And that would be the juice inside of a steak. If you're a vegan, that would be the juice inside of a zucchini. Just kidding. Okay, so those are saturated fats. Unsaturated fats, which are good for you. Unsaturated, why would they use oleic acid? Oleic acid is, I'll explain to you, it's the cure. Oops, sorry. oleic acid is actually the cure for ALD, the oligodystrophy, because they found that by having that, it actually ate up all of the extra fats that these kids were having in their bloodstreams. So this unsaturated fats means this is not saturated with the most number of hydrogens that you could possibly have. If you counted the number of hydrogens, these little gray balls right here, if you counted the number of these versus the ones that were in our saturated fat, this has more hydrogens than this unsaturated fat. And the reason is because we have double bonds in here. And by having double bonds, those carbons double bond to each other, and that causes this crink in the fatty acid chains. You get this weird little crink. Now they can't fit flat right on top of each other. They have this double bond, and we're not as structured, so we don't have the same structure. So at room temperature, we are liquid. So unsaturated fats are liquid at room temperature. You can do safflower oil, olive oil, EVVO. Yeah. What are some other oils? I can't think of any. Vegetable oil. All of those oils are liquid at room temperature. They can't be solid because they have carbons double bonding together, and once they've got double bonds, they don't need the extra hydrogen to complete that bond. So some of the hydrogens get kicked out. And this actually makes it easier for your body's enzymes to come in and break this down. When they come in and break this down, that makes it super easy to not have these stuck in your arteries, and therefore unsaturated fats can get broken down easier. They're a weird shape. They don't get stuck in your arteries, and this is much, much more healthy for you than is the saturated fats. Saturated fats are big, they're bulky, they can't bend, and they tend to get stuck in the arteries. Okay? All right. I just went over all that. So I will say this, that what we do, oh, we don't have a picture of it. So I will say this, that what they do with, let's say, fake butter. So fake butter, I don't even know what that's called. I can't believe it's not butter. That's it. So fake butter is actually bad for you. So I'm going to go back and give a disclaimer on that statement. But so it is like this. So it is a solid. So it's this kind of an oil. So it wouldn't be as good for your heart. So people who want to be health conscious say, I want to have something then that's like this. But this is not very easy to spread on your toast. This is not easy to, it's just not as convenient. So what they did is they came up with trans fats. Trans fats are where they actually put a trace mineral or a metal in here. And by putting a metal in, it bends it back and it makes it more straight. So now it will be just like butter. It will be solid at room temperature. Almost. If you've ever tried any of those, I can't believe it's not butter type of stuff, then it's almost solid at room temperature. It's just a really soft solid is what it is. If you eat peanut butter and you have what's the word here? Hydrogenized. There we go. Hydrogenized peanut butter. That means they didn't want you to have oil at the top of your peanut butter and you have to mix it in. So they took your peanut butter, which has some fats that look like this, and they actually put in metals and different elements in here to try to straighten it out. And now they call it no stir peanut butter, because now it's all going to be solid at room temperature, just as if it came from an animal. It's peanut butter. It didn't come from an animal. We know that. So, but they're trying to make it like that so that it will have that type of structure, and you'll be able to use your knife instead of pouring butter onto your bread, which makes it just kind of a little more awkward. So that's saturated and unsat. Always think fat is bad. So unsat is better for you. Sat is not good for you because of this consistent, no double bonds, and it's so saturated with hydrogens. But that being said, to be eating these trans fats is really bad for you. Trans fats are not a positive. It is super hard for your body to break them out. So are you better eating some fake butter that's supposed to be good for you but really isn't, and it's got trans fats in it? You should just go back and eat this. If you need a solid, go back and eat butter. All things in moderation. That's just Miss Brown's little piece of advice for you, free with this little video. Okay, we're getting towards the end because I'm getting tired. Fats, most animal fats, so anything with steaks, any of that are going to be saturated. Anything with plants and fish. Fish have unsaturated fats. You've never had it, but there's a thing called cod liver oil. Cod liver oil is an unsaturated fat and is actually good for you. Shark liver oil, which helps against cancers, is actually good for you. So it is unsaturated. Hydrogenation, we just got through all that. And fat is energy storage. So your fat, your adipose tissue, which is fat, sits in your body and holds energy in the form of fat. If you ever need it and you are stranded or on a reality show on an island, you can use that to get your energy until they come rescue you. Okay, moving on. So here's another type of fats. So these are fats, lipid, with a phosphorus group on them. That's all they are. So we're going to add a phosphate group attached to that glycerol, which is at the head of the fatty acid. And we're going to have 2 fatty acid tails instead of 3. And now we have a phospholipid, which is what makes up all of your cell membranes. This is what makes up the cell membranes. So this is the cell membrane. There's the 2 fatty acid chains. Are they liking water or not liking water? Oh, they're hydrophobic. They don't like water. Remember, they're nonpolar, so they don't want anything to do with water. So they automatically, this is so cool, this is fluid. This is not set in stone. This is not solid. They automatically turn all their tails inward. That way they're with like material, and they're not hanging around with water, which they don't like. So there is no water in here. This moves, and they say, I read the number once, it was something like in a minute you have 1,800 moves with these phospholipids every minute in each of your cells. This head might move to this spot, this one might move here, this one might go under and then come up right there. These things are constantly moving as they make your cell membrane. There's also other things in here that allow, because this is fluid, it's pretty easy. If you can get through this outside head, which likes water, is hydrophilic, and get in through here, you can actually have stuff go through the membrane straight into the inside of the cell. Okay, now I'm going to go back. So going back, choline. Choline is, actually ask me about that in class, I'll tell it to you. Choline is a molecule that helps in metabolism and helps, it's a metabolizer. I'll explain it to you when in class. Phosphate group, we're going to add a phosphate group to the glycerol head. So the phosphate is attached to the choline. These two grab onto a glycerol. The glycerol is going to have 2 fatty acid tails, just 2 of them, and now we've made the component of all the cell membranes in your entire body. So, and we call it a bilayer, so it's called a phospholipid bilayer, which I think is pretty evident right here. It's a bilayer. There's 1 layer, there's 2 layers, so we have 2 layers. Phospholipid, phosphate is in the top. Lipid is right here with the glycerol and the fatty acids. And this is what we end up with to make all of our membranes, all of our cell membranes. Now, like I said earlier, there are other molecules in here. Sometimes there are huge proteins, which we'll go over later, that actually sit like almost like a big tube just sitting in between this. And that tube allows bigger products to be able to go through and get inside of the cell itself. There's also other components that are within this phospholipid bilayer, and some of those are steroids. So steroids we'll talk about in just a minute. That's going to be 4 fused rings. Steroids are the fossilized version of a steroid, is how we know when cells first started producing proteins, which is literally 2 billion years ago. We found fossilized steroids. And then cholesterol. Cholesterol is really important. So cholesterol helps to keep the phospholipid bilayer from stiffening up. So it keeps it fluid and lets it being able to move. So too much of it can be bad because it gets stuck in your heart, but you need this in your in your phospholipid bilayer. So this is a steroid. Anytime you have these 4 rings hooked together, that's what makes a steroid. You have all kinds of steroids in your body. A steroid is a communicator molecule that allows one part of your body to communicate with the other part of your body. This will carry with it some communication, and we'll do that later in the semester. And proteins. So we're done. So that's what it looks like. And then whenever you see cholesterol, you're going to see little pieces of cholesterol in here. And that cholesterol helps keep this fluid. Cholesterol, though, is an animal fat. So if you get too cold, it literally will make the phospholipid bilayer get harder. So the body has to be kept at a certain temperature in order to keep the cholesterol fluid. If it gets too cold, then this will actually stop being fluid. You will not have that moving around, which can be bad because that may not allow you to take in nourishment or minerals or trace minerals that need to come from outside the body inside the cells. So you may not be able to have that. All right. I don't know how long that was, but that was long. So that's it. That is your lipids and your carbohydrates. I will do proteins in a minute.